Welcome back to the Sand Science Experience, where we explore the most exciting new research papers and preprints just for you. If you enjoy diving deep into fascinating science, please take just a second to like this video and subscribe to the channel. It really helps support the Sand Science Experience. Lets us keep bringing you this stuff. Okay, so let's dive in. Trying to figure out health, especially metabolism, things like obesity, it can get pretty confusing fast.
Oh, absolutely. There are so many ideas floating around.
Right. And sometimes ideas that seem solid, like, just take more antioxidants for oxidative stress, they don't always work out, especially for chronic metabolic stuff.
Exactly. It's often more complicated than that.
And deep inside our cells, you've got the mitochondria, the powerhouses, right? They make these things called reactive oxygen species, mROS.
And mROS isn't always the villain. It does important signaling.
True, but too much of it, especially when the body's under chronic stress, like in obesity, that is harmful.
That's the problem, the excess.
So for years, scientists have been trying to nail down where exactly all this extra harmful mROS comes from in obesity. Because if you know the source,
you can target it much better.
Yeah.
Rather than just, throwing general antioxidants at everything, which often doesn't work well.
Precisely. So our mission today in this deep dive is to unpack some really interesting research sources you shared. They seem to have found something specific, maybe even surprising, about mROS production in obesity, specifically looking at the liver.
That's right.
What did they actually uncover? Where's this excess mROS coming from, and what's it doing?
Okay, so here's the really fascinating bit, the kind of core surprise from these sources. The extra mROS in obese livers, it's not just some general mess.
No.
It seems to be driven by a very selective mechanism. It looks like it's directly linked to an imbalance involving coenzyme Q, CoQ, and this specific process in the mitochondria called reverse electron transport. Reverse electron transport.
Okay.
And this particular pathway happening in the liver seems to be a really major specific driver of the metabolic problems we see in obesity.
Wow. Okay. So not just any old mROS floating around. It's a specific type made in a specific way. How does that work inside the mitochondria with this CoQ and RET thing?
Yeah, so think of the electron transport chain like this.
Okay.
This assembly line for energy, right?
Right.
Electrons usually flow forward through different stations, these complexes.
Right, to make ATP the energy currency.
Exactly. Complex I is usually the first stop. But what the sources show is that complex I can actually be forced to run backwards. That's the RET part.
Backwards, huh?
Yeah. When conditions are right, specifically involving that CoQ we mentioned, electrons get pushed back into complex I from further down the line. And here's the key. When complex I runs backwards, specifically at one spot on it called site IQ, it acts like a leaky valve. It just spews out way more mROS superoxide, hydrogen peroxide, than it does when running normally forwards. Significantly more.
And the sources found that this specific RET-driven mROS from that site IQ in the liver was the main thing ramped up in obese mice.
Precisely. They looked really closely. They checked like 11 different possible places mROS could come from in the mitochondria of lean versus obese mouse livers.
11. Okay, thorough.
Yeah. And out of all of them, only the mROS made by RET, specifically from site IQ, was consistently and significantly up in obesity. It wasn't just leaky pipes everywhere. It was that spot.
Okay, so this specific backward process is the culprit in those mice. What causes that CoQ imbalance you mentioned? Why does the system suddenly start running in reverse like that?
Well, the research points pretty strongly towards the liver not making enough CoQ properly during obesity. CoQ has two forms, oxidized and reduced CoQ and CoQH2.
Right.
They found lower total CoQ in the obese mouse livers, sure, but the critical thing was a big jump in the ratio of the reduced form to the oxidized form. So much more CoQH2 relative to CoQ.
Ah, the ratio is key.
Exactly. That high ratio of the reduced form, CoQH2, it's like building up pressure. It provides the thermodynamic push needed to shove those electrons backward through complex I, driving RET.
Okay, that makes sense. Too much reduced CoQ backs things up, forces reverse, and boom, mROS from site IQ.
And they even found the genes responsible for making CoQ were turned down, suppressed, in the obese mouse livers. That explains why they weren't making enough and why the ratio got skewed.
Got it. So this specific RET-driven mROS, does it actually cause the metabolic problems, or is it just happening at the same time? Correlation versus causation.
That's always the question, right? But the sources give some pretty strong proof for causality.
Oh, yeah. How?
Well, they did experiments where they specifically made ER-Hardy errors happen just in the livers of healthy lean mice, using some clever tools. And guess what? Those mice developed problems with glucose tolerance and how their livers responded to insulin. basically mimicking some key features of obesity's metabolic mess.
Wow.
And then they did the opposite. In obese mice, they used genetic tricks to stop that mROS production.
Right.
Or even just gave them CoQ10 supplements, which helped fix that ratio.
Yeah.
It significantly improved their glucose tolerance and liver health. Better results in the obese mice. That points pretty strongly towards cause and effect.
Okay, that is compelling. But is this just a mouse thing, or does it actually matter for humans dealing with obesity, fatty liver, that sort of thing?
Super relevant, it seems. The sources also looked at liver samples from people with obesity and different stages of fatty liver disease, steatosis.
Okay, human data. Good.
And they found the same suppression of those CoQ-making genes in the patients with fatty liver disease.
Yeah.
Just like in the obese mice.
Really? The same pattern?
Yep. And maybe even more telling, that CoQ-H2 to CoQ ratio.
Yeah.
In the human liver samples, it's strongly correlated with how severe their fatty liver disease was.
Wow. So the worse the CoQ ratio imbalance, the worse the liver condition.
Exactly. It strongly suggests this whole mechanism messed up CoQ production, leading to a high ratio. Driving mROS is likely a big player in human obesity-related liver problems, too.
Okay, let's quickly recap this deep dive then. We started out with a general confusion, excess mROS and obesity, but we learned it's surprisingly specific.
This CoQ imbalance driving reverse electron transport, RET, specifically at complex I site IQ in the liver.
Right. And we saw pretty strong evidence in mice that this specific process actually causes metabolic issues like poor glucose tolerance.
And crucially, there's similar evidence showing up in humans with fatty liver disease, linking back to that same CoQ pathway.
So what's the big takeaway, the significance?
Well, it's huge, potentially. By identifying this specific source, this mechanism, researchers aren't just talking about vague antioxidants anymore.
Right, it's more targeted.
Exactly. They've pinpointed real molecular targets, maybe boosting CoQ production in the liver, or finding drugs that directly block RET right at that site IQ. It opens the door for potentially much more effective therapies down the line.
That makes a lot of sense, moving beyond the general approach.
And here's maybe a final thought, something the sources kind of hinted at for you to chew on. CoQ is already a popular supplement, right?
Yeah, lots of people take it.
But the researchers suggested that maybe current supplements need, well, reimagining was the word used, to actually get delivered effectively to the liver mitochondria, where this specific problem is happening.
Wow, delivery is key.
And they also floated the idea that maybe this CoQ arrate mechanism could help explain something else, why statin use, which can affect CoQ levels, is sometimes linked to a higher risk of type 2 diabetes.
Oh, interesting connection.
So it makes you wonder, right? What could a really targeted approach focusing just on this specific liver pathway mean for future treatments, or even how we think about the supplements people are taking right now?
Definitely food for thought. Really fascinating insights today. Thanks for walking us through that. And thanks to all of you for joining us for this deep dive. Please don't forget to like this video and subscribe to the channel for more explorations into the latest science. It genuinely helps support the Sans Science experience. We'll see you next time.